Welcome back to ACER Explained. I'm going through the Section 3 part of the ACER Practice Question booklet, so grab it so that you can follow along, and yeah, we'll be going through Unit 7 in this one. So Unit 7 goes through questions 16 to 18, and as usual, I'll be identifying some prereq knowledge, reasoning skills, and bootcamp resources that you can use as well. Unit 7 gives us some information on Budgerigar feather colour, and it says that the colour is determined by two independently inherited genes. Okay, and the first gene looks at outer layer pigment, and the second gene looks at pigment in the core. And importantly for question 16, we have two independently inherited genes. So these two genes are independently inherited. Okay, that's important for question 16 because question 16 is asking us about the genes. It's saying that the genes for the two pigments, would they most likely A, differ by one base pair, B, be on different chromosomes, C, be activated by the same mRNA, or D, have the same number of base pairs. All right, so we're looking at the information that's given to us. And now we're not given any information about the actual kind of codons in this gene or the actual base pairs. So if you have kind of previous knowledge about genetics, you can infer that it wouldn't differ by just one base pair. Two different genes typically differ by a lot more than just one base pair, so this would be incorrect. But if you didn't know that, I would put a question mark here because we're not really given that much information regarding this in this stimulus, okay? So then I'll move on to the next one. Would they be on different chromosomes? Now, if I think of the basic genetics knowledge that I would have in terms of prerequisite genetic knowledge, you'd have learnt about meiosis and the law of independent assortment. And so the law of independent assortment basically says that the chromosomes can be arranged independently of one another in meiosis, and that is a source of variation in your offspring. Okay, so if these genes are on different chromosomes, they can be independently inherited. If a gene was on the same chromosome, so if gene A and gene B, for example, were on the same chromosome, they will be inherited with the same chromosome, okay, during independent assortment. They wouldn't be separated. They could be separated through recombination, but during independent assortment, they are most likely going to be together, especially if they're closer together on the chromosome, because they would avoid recombination there as well. Okay, if they're on different chromosomes though, so let's say you have A on this chromosome and B on this chromosome, it doesn't really matter where A goes. B doesn't have to follow A. B could go in the opposite direction. It could, they're inherited independently in meiosis. And so this is a pretty good candidate for the correct option, right? Because we know that they're independently inherited. So then if they are independently inherited, they're more likely to be on two different chromosomes. Either that or very, very far apart on the same chromosome so that they're not impacted by recombination. Then we have C, be activated by the same mRNA. So you don't really need to know anything about genetics. If it's saying that it's activated by the same whatever, if it's activated by the same blah, you know it's not independent. They are dependent because that means that if you have more of this mRNA, then you're not only upregulating or you're not only increasing A, you're also going to be increasing B because they're activated by the same molecule. So that means that they're not independent. They are dependent on each other. So this one you can pretty confidently eliminate. Then you have, do they have the same number of base pairs? Again, if you know your genetics, you know that this is irrelevant to independent inheritance. So you can eliminate this. But also, there's no information really about the number of base pairs in the stimulus. So you could put a question mark. But because we're already relatively confident that B is the correct answer, we can eliminate these two and be fairly confident with the answer option B. And there we go, B is the correct option. So for this question, it is helpful to know your genetics, so to know your meiosis and the law of independent assortment, because that would be able to help you really quickly pick out B as the correct option. The reasoning skills used are identifying important information, as well as the process of elimination here if you weren't too confident with your genetics. So you had to kind of recognize the important information here was the fact that it's independently inherited. So on the screen, I put it there because I already picked it out as important information. But when you're looking at the stimulus, there's nothing that really points to that. You have to be able to read the question and link that to the independently inherited genes piece of information that's given in the stimulus. So the Bootcamp resource for identifying important information, that's a reasoning skill. So we have the reasoning skills course. Then in terms of genetics, genetics is pretty high yield in the GAMSAT. So I would definitely recommend knowing your basics for genetics. And so in the bootcamp, you can brush up on your genetics with the bridging course, genetics, as well as the content in action, genetics 1 and genetics 2. So yeah, that's question 16. Question 17 is asking us about a Punnett square, essentially. Well, it's asking us to create a Punnett square. It's asking us what the phenotypic ratio of this particular cross would be. And in order to find out the phenotypic ratio, you need to find out the genotypic ratio. And for this, you need to do a Punnett square. So first of all, what you would start off by doing is by separating these out, okay? So the gametes that you would get from this individual here would be capital Y, capital B, capital Y, lowercase b, lowercase y, capital B, lowercase y, lowercase b. And then the gametes that you'd get from this individual is lowercase y, capital B, lowercase y, lowercase b. And it would be the same thing again because the other one is also lowercase y, capital B, lowercase y, lowercase b. Okay? And you have to cross these gametes. So you have Y, B, Y, B, this one here, again, and this one here. You cross those with capital, then lowercase, lowercase b, okay? So you have capital Y, lowercase y, and then two capitals here. So just filling this across. Okay, and continuing to do that. This is a bit of a tedious process, but with practice, you can get faster at it. Lowercase y, B, B, you're just matching the rows and columns there. And then again, over here, you have, lowercase y, capital B, and then B, lowercase, 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 capital lowercase, and then same here. Okay, so now you have your genotypes here, okay? All right, and from the genotypes, you need to figure out what the phenotypes are going to be. Okay, so the information that we're given here is that the dominant allele, capital Y, is going to produce a yellow pigment, okay? And capital B is going to produce a dark pigment, okay? So when you have, and when you have the lowercase, there's no color. Okay. Then we're given some examples of different combinations. So when we have a yellow capital pigment mixed with a dark capital pigment, so the two dominants, we have Y and B, okay? And that creates a green feather. Then when you have no pigment on the outside, so you have a lowercase yellow, okay? So two lowercase yellows, because if you had one capital, because it's dominant, it would be the same as this one here. But we have two recessive alleles, so that means there's no color. So I'm going to put that there. So it doesn't really matter what's here. If it was another capital or a lowercase, it doesn't matter. But here, they should be both lowercase to get no color. And you have a capital B, you would get a blue feather. Okay? And then the answer options were also given yellow and white. Okay, so we can say that the yellow one, we can infer that yellow anything plus no color would lead to yellow. And then none of either would lead to white. So we have now our 4 phenotypes here. Okay? So now we have to figure out the ratios of green, blue, yellow, and white phenotypes. Okay? So I'm going to start off with green. Okay, so we're basically, for green, we're looking for 2 capitals. So we have 2 capitals here, so this is green. 2 capitals, green. We have 2 capitals, green, green. This one's green. This one's green. We have, that's it for the green ones. Then we have blue for those that are capital B, but both lowercase y. So we have blue here, blue, blue, here as well, and here as well. And then finally, well, we also have yellow for when we have capital Y and lowercase b's. However, we don't, oh, there we go, we have one over here. Capital Y and lowercase b. And then we have white. So that's going to be these ones. Okay? So now we have 2 white, we have 2 yellow, we have 6 blue, and we have 6 green. Okay? So we can simplify this to say 1 white, 1 yellow, 3 blue, 3 green. And this ratio is seen here, 3 green, 3 blue, 1 yellow, 1 white. And there we go. That's the answer. And you'll get better at kind of doing Punnett squares with practice and just taking a look and being able to decipher the phenotype more quickly. But these are kind of more tedious questions. So it might be something that you skip and come back to later. For this question, you did need the knowledge of how to do a Punnett square. And the reasoning skills that you had to use were, first of all, ignoring irrelevant information, but also picking out important information. So looking at the diagrams and seeing that the capital Y and capital B meant green, the lowercase y and capital B meant blue, and then inferring that with only a capital yellow, you'll probably have yellow, and with only lowercase for both, you would have white. So just a little bit of inference there as well. So the bootcamp resources here are the same as before: reasoning skills course and the bridging course on genetics, as well as the 2 content in actions for genetics. This question is asking us about fraternal or non-identical twins. So fraternal twins are essentially the same in terms of genetics. They're equivalent to siblings, okay? So this is why I've brought back the Punnett square that we drew earlier here, because we have this great example of parents and all of the potential offspring as well. Okay, so We can use this to eliminate the options here. So it says the offspring cannot have the same phenotype. This is not true because you can tell over here by the amount of greens and the amount of yellows and blue and white options, well, offspring. There's multiple potential offspring that have the same phenotype, and so they can have the same phenotype. So this is not true, right? Because this is saying they can't, but they can. Then we have the offspring cannot have the same genotype. We take a quick look and we see that we have multiple offspring with the same genotype. So they can have the same genotype. It's probably rare, but they can. So this is wrong. Then we have the two offspring and two parents could not all have different phenotypes. So it's saying they can't all have different phenotypes. But we can see here that, for example, if you have this one and we have this one, and then let's pick at random even, like let's pick this one here. That's different to both of the parents. And then let's pick another one. This one here is different to both of its parents. So they can have all different phenotypes. So then this one is incorrect. Then we have the offspring could both have the same phenotype and genotype as one parent. So let's take, for example, let's take this parent, okay? One of the offspring we can see here, this offspring has the same genotype as this parent. And then same thing here again, has the same genotype as this parent. So this is true. And so we can pick D as our correct option. And there we go. D is the correct option. With this one, you had to use kind of your Punnett square. You could have done it without the Punnett square as well, just trying to reason through it, but the Punnett square would definitely have made it easier. As well as the knowledge of fraternal twins. You could have inferred that as well because it does tell you that they're non-identical. So you can tell that it's going to be genetically different. It's not going to. It doesn't necessarily mean that they have to be genetically the same. And the reasoning skills used here is, again, ignoring the irrelevant information and focusing in on the relevant information. And the bootcamp resources that can be used here are the same as before, all the genetics courses for bridging course and content in action, as well as the reasoning skills course for the process of elimination, which is another reasoning skill that we used for this question. So that's the end of Unit 7. So again, like, share, and subscribe for any more GAMSAT help. We really love the feedback. And we have our Instagram here at ThankFlipGAMSAT, as well as our podcast, ThankFlipGAMSAT Nurses Doing GAMSAT, if you want to hear more from us. See you in the next one.